Maybe we'll get started. Okay, hi everyone. Before we get started, I'd like to acknowledge the traditional custodians of the lands on which we meet today. So Melbourne, that's the Wurundjeri people of the Kulin Nation. That's land that was never ceded. I would like to pay my respects to their elders, past and present, and extend that to any Aboriginal and Torres Strait Island people joining us today. So today's seminar is all about that road to independence in academic research. Many of us have aspirations of one day running our own lab. So today is really an opportunity to hear from three researchers about how they got to where they are and some of the lessons they've learned along the way. And also to ask any questions that you have about that process. So there'll be plenty of time at the end for questions to any of the speakers. You can feel free to start putting them in the chat, or alternatively, you can raise your hand by opening the participants. If you click on participants, down the bottom, there's an option to raise hand, and I can call on you and you can ask your question directly. Yeah, so up first we have Dr. Natalie Wee. She's our newest recruit to SVI and the most junior member of our panel. Natalie joined the Bone Cell Biology and Disease Lab this year for her third postdoc after being in the US for a number of years. And she is one of this year's rising stars at SVI and the EH Flack Fellow. So I'll leave it to you, Natalie.
Okay. All right, can everyone see my screen? Yeah. All right, cool. So I grew up mainly in Sydney, and I was always fascinated by science and medicine, and that's why I pursued research. I did my Bachelor of Medical Science with honours in physiology at the University of Sydney, and then a PhD in bone biology at the Garvan Institute. So today I just wanted to focus on a little bit about choosing a postdoc role and finding your path. And I think the two main things that I like to keep in mind is to tailor it to you and your needs. So not all postdoc positions are the same, and it's important to find the things that suit you best. So find a research niche, so an area where you can set yourself apart from other people. Consider the techniques that might be involved, the support available in terms of supervision, mentoring, resources, and also how it aligns with your personal circumstances or work-life balance. And so for me, one of those factors was location. Where am I in the world? And the second factor that I think is also really important is that a postdoc position is different from a PhD. Although you're still doing research and you're involved in a project, but the balance it shifts a little. And so an important question is, what do I have to offer and what will I gain from this experience? And it's important to consider a postdoc position as an exchange of skills and knowledge. So when I first began searching for my first postdoc position, I was very interested in the neural regulation of bone. It was an area of research that I was really passionate about, but there were very few labs in Australia involved in this type of research. And so I took my search overseas and focused on looking at labs in the US. I began searching in September 2015. And so this was about a year and a half before I finished my PhD itself. And I began looking at the biggest meeting in my field, which is the American Society for Bone and Mineral Research. I think it's also important that you look at different ways of finding a postdoc position. So I had introductions set up by my PhD supervisor to meet other potential supervisors. I also did a lot of interviews and networking at conferences. And I also emailed out to labs where I was interested in their work or interested in working with a particular supervisor. My first postdoctoral position was in the skeletal neurobiology lab at Washington University in St. Louis. So I originally met my PI there at a US conference, and then followed up with multiple Skype calls, and was subsequently invited as an invited speaker to visit the campus there and give a talk, and was offered a position. And this lab was a newly established lab, and it was focusing on the innervation of the skeleton, which is the area that I really wanted to work in. They were looking for someone with experience in bone and neuroscience, And the lab was focused on developing new techniques for imaging. So this really worked well for me because it had a strong histology aspect, and that's a skill set that I had worked quite a bit on it during my PhD. I saw a lot of potential in this lab. The supervisor there was very ambitious and had a lot of great ideas. And in general, the institute itself had a very strong bone biology program. So there were at least over 20 groups involved in bone research. And they had a very strong trainee program and a lot of guest speakers. And so it led to a lot of exposure to ideas within the bone biology field. And there were also opportunities for student supervision while I was there. After about 8 months, I actually reevaluated my needs and my fit for the position. And I decided to look for another postdoc position because I thought that I needed something a little bit different. But while I was there, the research project itself was incredibly exciting, and I had made some really large contributions to that project. So although I was there for a short period of time, I continued to collaborate with them for the couple of years after I had moved on. My second postdoc position came to me by chance, that I'd received an email from my PhD thesis examiner saying that he was looking for a postdoc with my particular expertise. I subsequently interviewed with him and was offered the position and took the position on in August 2017. And this was quite a stark difference between my first and my second postdocs, because this lab was a little different. It was quite a well-established lab led by a senior PI, and his expectation was that the majority of my time was focused on our individual research projects. So I spent a lot of time in the lab working towards papers. I didn't take on as many students for student supervision. So it was quite a different balance in what my role was as a postdoc. And this worked really well in terms of building my track record and building the techniques that I was exposed to. Okay, so during my second postdoc, so I was in my second postdoc for about 2.5 years. So my major project, which is why they hired me, was to look at neuropeptide regulation of bone fracture repair. And then I took on a second project looking at a sensory neuropeptide and how it regulates the skeleton. But I think what was really interesting and unexpected from this postdoctoral position that has actually really shaped my career, my subsequent career steps, is that I actually assisted on another 2 projects in the lab. And so I did some histology for particular projects that were fundamental to that lab's research. So the lab was very focused on lineage tracing. And that led to a couple of co-authorships, and it shifted my focus. And I got really excited about lineage tracing within the skeleton, and subsequently took on a third project and looking at lineage tracing of a progenitor cell population within the cortical bone. And this was really interesting because coming out of my PhD, I don't think I realized that during my postdoc, I can kind of shift my research direction a little bit and try new things without deviating too far from where I want to be. So I gained a lot of additional skills and exposure to these new techniques. And that's really shaped where I am now, because I subsequently decided that I wanted to move back to Australia. And so coming back to focusing on what my needs are in terms of what's a good postdoc position and where do I want to go, I actually changed what I was looking for. I focused more on a position where I could begin to transition more to become more independent. Somewhere that would foster and develop my own ideas. And I would look for a PI in a lab where they had a complementary skill set which would really work well together with what I could offer. Since I had been working in the bone research field for about 6-7 years at this point, I'd met a lot of colleagues, and so with this particular Sept, I relied a lot on personal recommendations. And in July last year, I contacted Natalie, and we subsequently met up, and this led to us developing project ideas, and I began applying for grants and fellowships, subsequently leading to me obtaining a fellowship and rising staff, which supports me here at Simpson's Institute. And reflecting back on my postdoctoral positions so far, is that they've really shaped where I am now. So it's given me a skill set in lineage tracing and progenitor cell populations, which is somewhere I had not expected that I would end up, because I thought I was going to follow more neuroscience initially, but this is something that I'm incredibly passionate about. I've continued to build on the skill set that I have. So in every position, I have continued to focus on histology techniques, and histology of bone is quite specialized and requires multiple techniques. And I've been incredibly fortunate that I've been able to learn all of them. So frozen, plastic, and paraffin. And so that's something that really does tend to set me apart in job applications. Here, I was looking for a complementary skill set. So while my focus is understanding rare cell populations within bone that have regenerative potential, by joining Natalie's lab here, it was really a great opportunity, because a lot of bone research focuses on bone quantity, but Nat's perspective on the bone research is quite broad, and she looks at the structural integrity, the makeup of the bone, its organization. And a lot of these factors all contribute to bone strength, which is really what, in a translational perspective, we focus on, is how strong is the bone and will it resist fracture? And so I can see a lot of potential in terms of the grant applications and the type of research and the direction that we can do together, which makes this current position a really good fit for me. And also, on the path to independence, like where we have to build our track record. And so over the time, each of my positions, I have focused a lot on my publication record. And something that's been very important to me is making sure that whatever I begin, I see to the end. So I constantly see to publication. Sometimes it takes a little longer and it requires perseverance and patience. So one of my PhD papers only just got published this year. And so I've spent the last couple of years chasing it up, finishing it. And that's really important, I think, in terms of transitioning from like PhD to postdoc, is that, you know, what you begin, that you see to the end, and that you deliver on what commitments you make, because I think that's largely contributes to success. And then also another large part is that really we don't do anything by ourselves. A lot of the success that I've had is due to the opportunities, the recommendations, the advice, and the support from my supervisors, mentors, and colleagues. And so they really shape where I am today. So what's next in terms of what am I looking at and where do I want to go? So I'm really interested in establishing my own research direction, building collaborations, supervising students, doing a lot more grant writing, and working towards having my own lab. And then, so I guess my take home from it all is so what makes a good postdoc position? And really, good is quite arbitrary. And I think it's important to be specific. Know what you want and know what you need and what's going to help you along the way. I think a good position is something that works to your strengths, improves your weaknesses, and broadens your perspective. And so what's good for one person may not be good for another. And so I think it's really important to make sure that you find out what's going to suit you. And I think something that is important everywhere is that you have a very supportive environment, whether it be your colleagues, mentors, friends, I think they all contribute to your overall success. In terms of searching for a postdoc position, it's good to look early and to keep your options open. And I think your chances of obtaining a position are much more favorable when you have something to offer the lab in terms of your skill set or your knowledge, or alternatively, if you could source your own funding through a fellowship or a grant. And then finally, just my acknowledgements to each of my supervisors in each of my positions, and then also to my many collaborators, colleagues, students and friends that over the last couple of years. Thank you.
All right, thanks, Natalie. It was really, really awesome for all of us to get to know you and what your background is, but I think some really great advice, particularly for some of the PhD students who are looking for postdocs right now and setting up that, so thanks. Next, we have Davis McCarthy, who started the Bioinformatics and Cellular Genomics Lab at SVI in 2018, following a very successful PhD and postdoc in the UK. I think we all know that his story is one of rather meteoric success, transitioning quite quickly through the ranks, and I'm looking forward to his insights from someone who has recently started his own group.
Thanks, Jackie. Thanks for the chance to speak. So my plan was to talk through my path, and I have one slide with some thoughts. Jump in at any point if you have any questions, and of course, happy to discuss anything either now or after the presentation or offline later. So hopefully that is working. So I'm from Melbourne. I grew up here, went to school here, and I started off at the University of Melbourne. I spent time at the Walter and Eliza Hall Institute, as I'll discuss in a second. Went on to PhD or DPhil at the University of Oxford at Balliol College there. Did a postdoc at EBI, the European Bioinformatics Institute in Cambridge. And then, as Jackie said, end of 2018, I came back to Melbourne to start a group here. So not much to say about the University of Melbourne. I did the ultimate degree for someone who leaves high school not knowing exactly what they want to do, which is a Bachelor of Arts, Bachelor of Science combined degree. So I studied German in my Bachelor of Arts. That was a real passion of mine. That was great fun. It took me to Germany on exchange. It took me overseas several times. It was a great experience, and I have no regrets doing it. Bachelor of Arts, I think, was also helpful for writing, which becomes a more and more important part of being a scientist as you continue in your career. And more importantly, though, for what I've gone on to do since then, I did a Bachelor of Science and majored in applied mathematics and statistics, dropping physics after first year when it clashed too much with my German subjects, and also required me to spend too much time in the lab. And maths is a great subject for people who are lazy like me, because you don't have to have extended practicals, you don't have to waste your time in the lab. And if you figure out how to solve the problems, then you can go and chill out. The most sort of critical time for me in the path that I'm on now was in 2006. I came back from 7 months exchange in Germany without a particular idea of what I wanted to do, but I happened to see an email go around the department advertising undergraduate research opportunities, and they were specifically looking to place people with a maths background in biology or specifically medical research. And so I thought that sounded like more fun than stacking shelves over the summer as a job. So I applied for that and was lucky enough to end up with a placement at the Walter and Eliza Hall Institute in the bioinformatics division there, working with Gordon Smythe. And so that was just a really lucky event, basically. And that introduced me to bioinformatics, to statistics, to genetics and biology as well. I hadn't studied biology since year 10 at school, so I had a fair bit of catching up to do, but with Gordon's help and other people at WeHi, I enjoyed it, got into it. And it is true that I started learning statistics with the Cartoon Guide to Statistics, and I started learning genetics with the Cartoon Guide to Genetics. I sat through a lot of seminars at Wee High, looking at facts plots that no one ever bothered to explain. So I spent a lot of time confused during that time. But with Gordon's guidance, I got into the area of gene expression and specifically developing methods for differential expression testing. So that started with thinking about microarray data. And so that's my first paper, this treat one there. And then when RNA sequencing started to become available, Gordon recognized that he had a lot of good ideas for how we might be able to analyze RNA-seq data effectively, and that led to the EDGAR project that he and Mark started, and then I was the sort of the third person to join, and that led to the EDGAR paper, their subsequent follow-up with more methods implemented in EDGAR, both the sort of the generalized linear model approach and then this quasi-likelihood approach. And so they were incredibly successful. The EDGAR paper has been cited more than 15,000 times. I can't take too much credit for that. I was in the right place at the right time, just kind of by fluke. But that was an amazing experience as a sort of an undergrad, and then in honours I got to do, and then as an RA for 18 months with Gordon afterwards, I got to do really genuine cutting edge research that happened to have an influence on the field. So that was an amazing sort of boost for me, and it opened up a lot of opportunities, specifically studying at Oxford for my DPhil, and again on the sort of the strength of what I was able to do with Gordon, got a competitive scholarship to go and do that. That was kind of a dream come true. Of course, in some ways I'd always wanted to do a PhD overseas, and that's the advice I'd received. So this was an amazing opportunity. It actually didn't go that well for some of it. So I got over there, and they were Peter Donnelly's group, my supervisor there, is very, very senior, famous PI, full professor at the Wellcome Trust Centre Institute. He was a full professor at the age of 29, so he's kind of ridiculous. He'd been a professor for a long time. He was involved in lots of stuff, didn't have heaps of time, to be hands-on with projects or guide students through things. So I was sort of thrown in there and shifted from the sort of gene expression space to statistical genetics, which was ultimately good, but it sort of threw me a little bit how much of a small shift in field could actually you feel like going back to the start. And so I also spent a bit too much time playing cricket and doing all the fun things you can do in Oxford instead of doing work. So I sort of had a sluggish start to my PhD, and along with some projects that didn't really work, I sort of actually suffered a bit of a loss of confidence in what I was doing for a while. In the end, it worked out kind of fine. I wrote one paper on variant annotation, which I found extremely boring at the time, but Peter pushed me to finish, and it's good that it did. It's a fine little paper. Then I worked on a very big consortium collaboration project on the genetic architecture of type 2 diabetes, which was very complicated, very challenging in lots of ways. And going back to Natalie's comment about finishing projects off, I agree, that is generally very important, but this sort of complicated collaboration led to a core of my PhD thesis, 2 big chapters on variance partitioning analyses in type 2, risk for type 2 diabetes, that I never actually published because it was just sort of too difficult, too complicated, and I ended up deciding in the end to kind of just cut my losses with that project and move on. The final thing I did in my PhD was what kind of led me out of the sort of disappointing aspects of the PhD and into stuff that I was a lot more excited about, and that was the SCADA project, a software package for dealing with single-cell RNA-seq data. And so that's kind of what reinvigorated my enthusiasm again, which was the arrival of single-cell RNA sequencing in Oxford, which sort of both took me back to the work that I'd really enjoyed with Gordon on gene expression, and then also was just super cool and something I jumped on as an exciting area that really seemed destined for growth, and also had loads of interesting statistical opportunities and ideas to explore. So that was how I ended up hearing about a guy called Oliver Stegler, a group leader at the European Bioinformatics Institute in Cambridge. So I saw a video of him give a talk at Biology of Genomes, and I just thought, that's super cool. He seemed to have the right sort of balance of interests of statistical genetics, but also single cell RNA-seq. So I got into that, and so I just sort of emailed him cold, and I went over to EBI after my PhD and spent a really enjoyable and ultimately very productive 3 years with him. And so on the right-hand side there are the kind of my major postdoc papers where I'm first author or co-first author on different aspects of methods for single cell analysis, specifically linking DNA variation to single cell gene expression variation. And that work there really sort of gave me an identity, I guess, as a researcher, and an initial plan to pursue as a lab head coming back to SVI. Another really nice aspect of being at EBI was that I got to work on a lot of collaborative projects as well. And there are some examples of papers that came out of various collaborations with lots of interesting people. And so I was really lucky to be there. Ollie had an amazing lab, lots of fantastic scientists working on really interesting projects. And that sort of, again, really invigorating sort of research experience that, again, gave me a lot of confidence to build on and keep going and develop my own ideas for what I would like to do as an independent researcher in the future. That got me through to sort of August 2018, and then over that time, especially in postdoc, been in discussions with SVI, with Tom and Anne and others, developing the idea of starting a bioinformatics, a dry lab, computational lab at SVI. And so the outcome of those discussions was happily a return to Melbourne as a lab head and a group leader. And part of what we figured out, what we thought would be important, was to have a very strong connection to other computational researchers, specifically at the University of Melbourne in the Melbourne Integrative Genomics group. And so that's led to my current position as a technically 0.5 FTE at SVI and 0.5 at the university. And happily, again, since then, things have gone well. Been able to recruit a really fantastic team of people working with me now, and been able to build on those things that I started to develop with Ollie in Cambridge on single cell genomics, and also in more recent times get into the sort of medical imaging field through collaborations with Breast Screen Victoria and St Vincent's Breast Screen Clinic at the hospital. So that's kind of my story in a nutshell. And the final slide of ideas I wanted to put for your consideration is this. My wife's a lawyer, and she's also thinking about career development and all those things. And one of the partners that she works with at the firm said that there are 3 things required to make the step up, in their case, from a senior associate to a partner at a law firm, and they are need, quality, and viability. And I think that actually maps very well onto making the step into a sort of faculty level position in science. I think you need to be able to persuade whoever you need to persuade that there's a need for what you do. In my case, SVI recognised that themselves, that they wanted in-house bioinformatics. So there was a clear need for that. And I guess I was able to persuade them that I was able to produce work of the required quality. And then that I could persuade them again that it was a viable proposition, that we in collaboration would be able to have a plausible route to funding that group over the medium to long term. So I think that's key things when you're thinking about how you might sort of pitch yourself. for faculty positions and other opportunities. I think if you can persuade people of those three things, then you can potentially put yourself in a strong position. So, you know, the need is crucial. If you're say a CRISPR-Cas9 person and you're applying to a department that has 6 labs already established doing gene editing work, then doesn't matter how good you are, the need might just not be there. And so that's a sort of an important starting point. Quality is obviously crucial, and this is kind of, I guess, the traditional track record stuff. Hopefully it's also, the quality point does resonate with people, that it's not just the amount of papers you publish, but you know, high quality papers that really make a contribution to the field are more valuable than loads of little papers. And then there's the viability, and that's a tricky thing, and gets into the complexities of grant funding and all of that sort of stuff. So I won't talk about more there, but happy to discuss ideas beyond that, without that. So I think I'll leave it there and go back to Jacki and others for questions and so on.
Great, thanks, Davis. That was super interesting, and yeah, I really like that mantra at the end. Okay, so last up we have Professor Helen Thomas, who is head of the Immunology and Diabetes Unit and also Associate Director at SVI. Helen has been an established group leader for many years now, so I'm looking forward to her perspectives as someone who has her own group but has also mentored many others through this career stage. If you'd like to share your screen, Helen.
Thanks, Jacki. Yep, I can share my PowerPoint. Oh, you can see my notes, hang on. Okay, hopefully everyone can see that. So I'm not going to go into a lot of detail about my background. It's written there on the screen if you're interested. But I guess Jacki's already introduced me as the
Hang on, Helen. I don't know. I can't see your screen. Can anybody else? No. Oh, there we go. Oh, there.
Can you see it?
I can see it. Yeah, now I can see it.
Okay, and can you see the right one? You can see the full screen?
Yeah. No notes.
Cool. Okay. Yeah, so as Jacki said, I'm the head of the Immunology and Diabetes Unit at St Vincent's, which is actually a group of about 30 people. So it's quite a lot of people there, which is comprised of about 5 labs, and we're all broadly focused on the pathogenesis of diabetes with different skills like islet biology, autoimmunity, immunology, genomics, including mouse and human immunology. So in that group, I supervise and mentor students, postdocs, and the lab heads in the group, which is, obviously a much, it's pretty broad role. I'm also an honorary professor at the University of Melbourne, which doesn't really actually have any role attached to it other than a fancy title, and associate director of SVI, which in that role, I'm currently responsible for the Institute's gender equality portfolio, and happy to discuss that with anyone who wants to. So I guess I'm going to stress that this is a more than 25-year career that I'm going to talk about here as sort of in type 1 diabetes research. So it's quite different to the other speakers. So rather than focusing on how I got to this point, I'm going to focus a bit more on some of the highlights of my career and some of the things that I'm proud of doing or being involved with having done along the way. And sort of, I guess, give people a bit of thought about some of the ways that you can sort of keep your career going once you've got established. Because if you think about how old you are when you've finished your postdoc, and then you have to make it out to when you're 65, when you can retire, it's quite a long time, and you've got to keep, you've got to keep your productivity up for all that time. So these are some slightly different thoughts about that. So yeah, I finished my PhD in 2000 at the Wee High, and as you can see there, it was after quite a long time working as an undergrad, as a research assistant. So it's been a little bit different at the start to many people in that I did a lot of travel and work overseas before and during my PhD. But then I stayed in the same group and research topic after I finished my PhD. So that's something that I probably wouldn't advise students to do nowadays, because I think that the change of research environment is really great for collaboration and broadening your scientific education. And I also just put on the slide that I have had 2 children along the way, and you know that definitely is a career disruption, and I just want people to I guess think about the idea that, you know, at some point in your career you're going to have disruptions with having family and other things. So just a bit of background, my research has progressed from a basic understanding of how autoimmune type 1 diabetes occurs in a mouse model, and then more recently to human clinical trials over the past 25 years. So I started out doing a lot of molecular and mouse work. I made a transgenic mouse in my PhD and characterized a bunch of knockout mice. Over the years, I've, like Natalie said, I've tried to use up-to-date methods and develop expertise in some of the techniques that are required to study disease. Sometimes this has meant learning some difficult techniques and traveling to other groups to learn how to do things, and investing a lot of time in getting different technology working in the lab. So I've tried to use some of that time and expertise, investment in time and expertise, in learning new methods in the best way possible. And the first example I'm going to show you is in islet isolation. So during my PhD, I had to learn how to isolate islets from mice, which is actually a reasonably tricky thing to do. Isolate the islets from the pancreas. You have to inject some enzyme into the common bile duct using a dissecting microscope, and then take out the pancreas and digest it, and shake up the tissue to release the islets, and then run it over a gradient. And it's kind of time consuming. I used to kill 16 mice just to get 1 million cells back. And so the effort that I put into learning how to do that was really great, because I was able to study the disease pathology properly in mice. But it also meant that we were able to translate some of those techniques into isolating islets from human organ donors, and then to set up our clinical islet transplant program. And so a few things that have come out of our clinical program that really was built on that technical skill in the lab. So firstly, we've made some great connections with clinical collaborators, and there's an old photo here of some of the clinical collaborators with me and Tom, and actually one of our first islet transplant recipient is this lady here, Elaine. So that's been really great, because the clinical collaborators are obviously really important for doing transplants. But secondly, the success of the program has given the people in our group a sense that they're making a real difference to people with diabetes, because when they come in and isolate islets, it sometimes is in the middle of the night or over the weekends, and those islets are used to cure someone's diabetes, then I think that people really don't mind doing those long hours for that clinical difference. And I guess the third thing that's come out of the program is that it's linked me with lab members, me and the lab members, sorry, with other islet biology researchers around Australia and the world. The islets that we don't use for transplant are used for distribution of research islets, and the people in my group have a chance to collaborate with a lot of people around the world to do that research. Okay, and so then the second thing that I was going to talk about is another example of translation of my work, and this is the clinical trial that's about to get underway. In fact, we're going to do our press release late next week for this trial. So this is really a fruition of a couple of decades of my research, and this timeline on here really only starts in 2012, but actually the the basic research that went into this trial went on for much longer than that. So we're going to use this drug called baricitinib, which is a JAK-STAT inhibitor in patients with newly diagnosed type 1 diabetes, which is a world first for this class of drug in type 1 diabetes. And, you know, if it's successful, it could affect how diabetes is managed clinically. So it's pretty exciting. I'm very excited about it. And yeah, so to get it going, it's sort of been a pretty long and arduous journey. So I'll just go through some of the things that we've done. When we first worked out that there would be that JAK inhibitors would be a suitable drug for type 1 diabetes, we went and talked to Andrew Wilkes, who was one of the first people to identify JAKs back in the 1980s or 90s, I can't remember exactly when. And he suggested we approach some different pharmaceutical companies that were developing JAK inhibitors, which we did. We obtained one compound from AstraZeneca, which we did our preclinical validation on, and that work was then used to take forward to Eli Lilly, who gave us their compound, which is very similar in action to the drug we're going to use, called baricitinib. And we also met with the funding body at this point in 2016 to talk about the idea of doing a clinical trial and getting that funded. They introduced us to some people at AbbVie who gave us another JAK inhibitor. So now we've got 3 different drugs that we were able to test and demonstrate efficacy using all 3 drugs, which gave us a lot more confidence that the class of drug would be successful in type 1 diabetes. And we published our work using the preclinical drug AZD. Then Tom and I went on a roadshow and visited all these companies. We went to visit Eli Lilly, and we met up with people from the JDRF in New York and talked about the idea of doing a trial. And then we got the investigator-initiated trial approval from Eli Lilly to use baricitinib last year. And at the start of this year, we received funding from the Juvenile Diabetes Foundation for the trial. And as I said before, the trial's going to start, well, we've actually got a couple of sites activated already to start recruiting patients as we speak. So to get this trial underway, a whole new set of skills had to be acquired by me and the group. And I can't say that I've done much of this anymore other than just sort of sitting at meetings and offer advice, but you know, writing trial protocols, getting HREC approval, working out site budgets, database, trial monitoring, and the list goes on. There's an enormous number of things that have to be, you have to learn to do to conduct a clinical trial. And it's not straightforward for someone who's spent 25 years doing basic research. And then this is the last example I was going to give, which is, you know, throughout my career, I've been supported by NH and MRC or JDRF fellowship funding. And when this ended in 2017, it was a really sad time for me because the competitive grant system just became so difficult and difficult to support people, you know, even people who I feel like I've, you know, made a strong contribution to research in Australia. So I looked at ways of diversifying the income of the lab so that we were not bound to receiving grants from NHMRC. And one of those things that we thought about doing was extending some of the pharmaceutical studies that we'd done with the JAK inhibitor. You know, the pharmaceutical companies were really happy with the work that we had done. We did a lot of pharmacokinetics, pharmacodynamics. These are terms used by drug companies to understand if their drug is found in the circulation and if it's hitting its target in vivo, and really important for clinical development of drugs. Sorry, I don't know if you can hear my son unpacking the dishwasher in the background. Sorry about the noise. So we received lots of positive feedback from the farmer about our work and decided to apply for funding from the JDRF Australia to set up a preclinical fee-for-service contract research organisation to do research for small biotech companies and pharmaceutical companies to try and accelerate some therapies that were being developed to type 1 diabetes so that we could get them through to type 1 diabetes clinical trials. This has really taken off in the last 12 months. So at the moment, we've got 2 projects worth about $400,000 and another couple of projects in the pipeline. But of course, it takes a lot of time to set this up, and it has required a lot of time from a couple of people in the group to get it up and running. But the additional funding that we get and the collaborative nature of the work that we're going to do has been really welcome. Okay, so now I've just got a few little pieces of advice. I hope I haven't gone for too long. So first of all, my one piece of advice is your publication record is still the most important thing to success. And Davis showed us his publication record, which is obviously pretty outstanding. So unfortunately, in the funding systems of today, where and how often you publish is really important. And so you need to look for opportunities to publish your work. As a student or a postdoc, it's good to have a safe and sure publication project or aim, and another that's high impact and high risk. If you finish your PhD or postdoc without a publication, it can really ruin your future career as an independent researcher. And you know, while reviews are good, it can't only be reviews. I think you need to have some primary author, first author papers out of your PhD, which should be achievable if managed well by you and your supervisor. Yeah, so that's one piece of advice. The second thing is to try and work with supportive people. So it's good to see Natalie Wee is working with Natalie Sims, and I too tried to work with as many people called Thomas as possible. This is just a really small selection of people that I work with, and I've mainly put this slide up because it's got this nice old picture of Tom and I doing an experiment in the lab back in Weehai. So I'm pretty lucky that Tom's very supportive of the work I do, and we work really well as a team. He's allowed me to progress from PhD student to lab head to unit head all the time while being a mentor. Tom tends to push me to do things that are a bit outside my comfort zone or that I think might be a bit too hard. But actually some of these things have been the most rewarding things that I've done. So for example, setting up the human islet transplant program or being head of the diabetes group. Neither of these things have been particularly easy, but they've both been pretty rewarding. And another thing is, if you work on a disease, is to always try and stay in touch with the goals to improve disease outcomes. So it's really important today to understand the nitty-gritty of how your molecule works in your mouse, but keep an eye on that big picture and how understanding that molecule will help people with the disease. And by doing this, you know, you should be able to progress your work through to clinical trials. And make fabulous connections with industry if that's what you want to do. And I've just got here a picture of, you know, type 1 diabetes used to be fatal before the discovery of insulin 100 years ago. And now, of course, it's a disease that's still most commonly diagnosed in children, and it's really important to remember that when we're trying to design therapies for type 1 diabetes. Oh, and there's like these come up. Okay. And that is all I have to say.
Great, thanks, Helen. That was super interesting. Actually, the speakers did a really great job and given me lots of food for thought. So yeah, if anybody has questions, they can either pop it in the chat, they can write in the chat if they want to ask a question, or you can put your hand up, raise your hand in the participant section. But maybe while people are thinking about that, I'll get started. So I guess maybe a general question. Is there anything that you wish you knew before you started your own lab, or that you'd better prepared for before making that transition to a lab head? I'm happy for anybody to start if they want to, or I'll just pick on somebody. Davis, maybe you want to start, since you've made that most recently made that transition. Might be more fresh for you.
Yeah, it's a good question. I think I tried to speak with people who'd done it before, and so there were certain things that had been flagged as potential problems that I was aware of. I think one key one was having kind of frank discussions or negotiations, if you want to call them that, with the host institution to try and start in a strong position. So having the resources to be able to make a successful start. And I think that's more important than ever, given the current funding environment. People can't just assume that it'll be right in the funding system. And so I was lucky again that Tom, initiating the discussions, so really asked me, like, what do you think it takes to set up a sort of a successful bioinformatics lab here, and really gave me the opportunity to tell them what I thought was necessary. And I'd heard sort of the horror stories of people accepting a faculty position because they were so pleased to be offered a faculty position, but then end up sort of hamstrung because it didn't come with enough support. So they ended up sort of using up their lab funds too quickly without being able to establish themselves. So I think I was lucky, and there was great support from SVI to allow me to do that. You can come back to me.
Yeah.
I might have more thoughts.
I think that's yeah.
Oh, I was just going to add something to that. Well, it's slightly different, but this, the transition from being a postdoc to being a lab head, one of the things is that you no longer get to do experiments as much. You have to manage people, and we're not really trained to manage people. So I think that those are the sorts of things. Learning how to let go of not being able to do the experiment yourself, and to trust someone in your lab to do that experiment for you, is really important. But also maybe asking for some sort of training in being a manager as well.
Yeah, I'd second that. And budgeting too. Like you're never taught budgeting as a student or postdoc, and then suddenly you're responsible for these budgets, and whether that's putting sensible things together for your startup, for grants, or just managing whatever budget you end up with. And so again, I think SVI has been good because we've got such a flat structure that can speak to Anne, can speak to finance, to get direct support. And I've really noticed a big difference. Having a foot at SVI and at the uni is a really stark contrast in direct support. SVI is very proactive about looking for opportunities for funding for people, and the grants office has been fantastic for me for getting grant applications in. And if I were solely at the university, I would have had none of that, and I could count 2 or 3 grants that have been successful that were sort of instigated from SVI, sort of, rather than from me. And if I'd only been at the university, I never would have thought to apply for any of those things.
Yeah, that's interesting. I can't see any other. Oh, hang on, here we go. There's a question in the chat. Natalie and Davis, did you take funding with you to your postdocs, fellowships, for example, and how important is this in your opinion?
Do you want to start, Natalie?
Okay. So my first 2 postdocs, both of my supervisors actually were incredibly well funded. They had about 4 grants each when I joined them. So I didn't bring any funding. They already had the support there. So I tended to focus more on my research. But obviously coming to SVI, it was a really important factor. So the fellowship made a huge difference to getting me here. And I think it's also changed like my position in the lab. I did a lot of research and a lot of lab work. And now it's kind of a bit more of a transition. Like, how do I manage like my fellowship or the grant funding? How does it help me generate like new results? And I think it's opening up different opportunities that I didn't really experience in my previous postdocs. And I think that's really beginning to help shape me for the future.
And for me, I joined Ollie's lab without any fellowship. He was very well funded, so that was great. He was able to say, come and join as a postdoc. And then he actively encouraged me to apply for things, which is, of course, on my mind too. So I applied for a Marie Curie fellowship with him. That was not successful, and applied for an NHMRC early career fellowship, CJ Martin. That was very helpful. It kind of didn't limit what I would have done with him. He had enough money to sort of employ me for 3 years, so that was fine. So I didn't change my sort of employment status with him as such. But obviously, especially in the Australian funding system, to have a track record of a fellowship, obviously hard to quantify exactly what a difference that makes, but one can only assume that it helps quite a lot. So that was good. And then also that formalized the link with SVI, since SVI was my Australian host institute for that. So even before coming back as a lab head, there was a plan to come back on the second 2 years of the CJ Martin. And again, advantage of that was it was flexible. So Ollie was able to fund me for an extra year beyond the 2 years from the CJ Martin, so I could stay on a bit longer and really take advantage of the environment over there before coming back to Oz.
Great. All right, there's a question from Michael Sharp. Do you want to just unmute yourself, Michael?
Hi all, it was very interesting. I just want to know, was there ever a point where any of you felt like falling off the academic bandwagon and go into industry or teaching or anything like that, anything that, a PhD could get you? And then if so, what kept you on, applying for grants and doing the typical academic cycle? What kept you in the game?
Many times, Michael, many times. Yes. Look, when you don't get your funding, your fellowship funding or whatever, that's a good time to start thinking about your career path and whether you think that you're doing, whether what you're doing is worthwhile. And I guess you can see from the examples I gave that really that made me turn the career into a slightly different path, potentially slightly more sort of translational and industry focused than academic, although, obviously still trying to keep the academic focus as well. So yes, I think for me, it was probably mostly based on funding.
Yeah, for me, I've sort of said to myself over the years that while I enjoy doing it, academic science, and while people will pay me to do it, then I'll stay in it. But I've tried to, at least for myself, think that it's not the only possibility and it's not the only good possibility either. So for me, industry jobs will look quite different from most other people at SVI. People who I went through with, took sort of data science jobs at Google and Amazon and places like that. And so the kind of more obvious industrial sort of application of my skill set would be in the tech industry. And so I've always found it more motivating to think about trying to work on biological and medical problems rather than optimizing ad algorithms. So that's, the pure interest has kept me going. I've been super lucky with funding, so I haven't yet faced a point where I've really had to make a difficult decision about toughing through something. So fingers crossed that doesn't come, but every senior academic that I've spoken to has faced that at some point in their career. And so I'll have a decision to make at that point as to perhaps, does the inherent interest and belief in the value of doing science outweigh potentially difficult personal situation that a lack of funding can give rise to.
And I think, like, initially I always wanted to stay in research, but along the way when I was like looking for positions, I kept my options open, and I did look at industry and other positions, and I still kind of evaluated them based on, well, does this fit what I want to do or where I want to go, and what's being offered in terms of where do I see this taking me. I've always kind of stuck more with the research. I think that's really where my passion is. But I would say that while I was working overseas in the US, I was really quite amazed by the amount of interaction there is with industry. And I think that really has changed my perspective on how I see research in general. And there's actually a lot of interaction between all of the different roles in terms of like whether you take something in industry. I know I have a lot of colleagues that have taken up grant writing positions, and they all have different merits, and they all kind of contribute to the overall research and outcomes. And so I think they're all very valuable. I think it's just finding what suits you best.
Thanks. I have one more question. It's a question of sort of a little bit related to, I guess, imposter syndrome or self-doubt, and whether there was a point in your career where you went, yep, I'm ready to start my own lab now, or whether it really was just a leap of faith at some point that you can do it, and sort of how that's gone, how that feeling has changed over time about your sort of self-belief and things. Has it got easier? Has it got harder? Helen, you want to start?
I mean, maybe it's a little bit personality related, right? So there are obviously some people that never have that, and there are others like me, for example, that still have that today. So I think at some point I guess you sort of, for me it's almost been you wake up one morning and you think, oh, I manage about 4 people, therefore I must be a lab head, kind of thing. It's been a slightly different journey for me, partly because the sort of structure of going from postdoc to group leader to lab head wasn't so rigid then as it possibly is now. But I think it is a little bit of a gradual transition that you kind of realise that you've achieved at some point in your life.
Yeah. What about you, Davis?
Yeah, and for me, the sort of the crisis point, or the biggest doubts, were at times during my PhD. I had such an amazing experience at Weehigh, and I'd sort of been completely cocooned. Like Gordon was basically saying, "Oh, here's an interesting problem, let's work on it," and there was no pressure to deliver anything. We kind of did the stuff, and it worked out amazingly well. And then I made the switch to the PhD, very different environment, very different supervisor relationship. And for the first time for me, projects that didn't work for various reasons, and then sort of being distracted by other stuff. And so had the variant feeling and discussions with my supervisor, the PhD was not going particularly well. I had to work hard to get it back on track. And so that was, going from this absolute high to then this low. And so that was when like, oh, well, is this something I want to go through? Like, if this is how it feels to do this, then, going back to that point about enjoying it enough to keep doing it, there were doubts there. I knew I had to finish it because that was never a question. And then, so there was both the sort of that reinvigorated academic interest, which for me was sort of the single cell world picking up steam and being just excited about participating in that. And then going on to work with Ollie, which was really brought the excitement back to doing research with him. Just a change in environment. The fact of completing the PhD obviously helped with that, but then the projects themselves were great with excellent colleagues in a nice environment. Still there, I felt I really felt the pressure of delivering good projects. And so the sort of imposter syndrome was a little bit ameliorated for me because I sort of started having the discussions with SVI. So I knew that there was sort of interest there. There were opportunities, and say, even if it hadn't been with SVI, ultimately I knew that I was sort of at least in the mix for those sorts of things. So that helped again with building confidence. And then I think the sort of successful completion of the big postdoc projects then I think gave me really developed the confidence to say, okay, yeah, well, we've done some new stuff, and there's, we just started this sort of area of single cell genetics. Okay, I can see how much more there is to do here. And then it's like, oh, well, good, that's a research vision that I can pitch to other people, whether that's to SVI or to grant funders or whoever else.
Yeah. Did you want to add anything, Natalie? Obviously you haven't made that transition yet, but
I think to a certain extent, like imposter syndrome kind of hits at all kind of levels. Even transitioning from like PhD to postdoc. And so like I moved to the US, I didn't know anyone in that city, started a new job. I at first I was a little bit overwhelmed, and I think it's interesting because for me, having a bit of that imposter syndrome and feeling a little bit insecure has kind of helped me to be like, well, what do I need to do to make myself feel a bit more confident? How do I put myself forward? And I think that becomes the strength. Is like when I start a new position, it feels a little uncertain. It feels a bit unstable. But then once you take the steps and you go through the process, you feel much better, you become much more confident, and you really begin to develop what you want to do and where you want to go. And I think it kind of helps to a certain extent.
Yeah, I think that's really good advice. I think that's a really good spot to finish it, given the time. So starting on a high. So thanks to our panel speakers. That was all really fantastic. And thanks to everybody for coming.
Thanks for organising, guys. It's good.
Thanks, Helen.
Thanks.
Was I noisy? Was my background noisy?